To start this session, I'm going to talk about understanding clonal distributions during human neocortical development, deciphered through brain somatic mosaicism. Mosaic mutations that occur postzygotically can serve as lineage recorders for different clones during development, but also shows the clonal expansion from disease-related phenotype as a separate distribution compared with the background, as well as those sporadic mosaic contributions from environmental exposure. Using those neutral mosaic variants as recorders, we're trying to understand key questions like, first, how are human neocortical lineages distributed during development? Second, what's the total committed progenitor population size of the early human cortex? And third, can normal mosaicism explain why focal brain developmental disorders are always limited to one hemisphere? To address those questions, we first extensively sampled all the 5 cortical lobes from both left and right hemisphere, as well as cerebellum, heart, liver, kidney left and right, from one index case. For each of the cortical lobes, we further carried out 12 punches from the large piece, and then extracted DNA from both the small parts and also the large remainers. After homogenization and DNA extraction, we sequenced all the 25 samples at 300x at a genome scale, and also called all the mosaic variants using a combined computational pipeline. For the candidate variants, we further quantified all the variants with massive parallel amplicon sequencing in all the 25 samples, plus different punches from different lobes, and also fluorescent activated nuclei sorting and single nuclei. After the extensive sampling and validation strategy, we identified 259 mosaic variants from the first index case, and of all the variants, 110 were only present in one single lobe. By comparing those lobe-specific variants, we actually find there are stochastic distribution differences in left and right, and from anterior to posterior. Assuming 2 variants that present in the same lineage, if we compare their allele fractions in all the 25 tissues, we should see a positive correlation. Whereas if 2 variants are presenting in mutually exclusive lineages, we should be able to see a negative correlation of their fractions in all the 25 tissues. In this cluster, we showed the correlation between each of the 259 variants between each other by annotating the tissue where the variants are detected and the left-right distribution. We actually can see that there is a strong clustering between the left and right hemisphere, and also from the anterior to the posterior in terms of different lobes. To study the distribution of different brain cell types, we first homogenized and stabilized the raw samples and then prepared for nuclei and used antibodies to do fluorescent activated nuclear sorting for major brain cell types like neurons, oligodendrocytes, astrocytes, and microglia. We use this lolly plot to show the distribution of allele fractions in different cell types and also in each of the different lobes. y-axis shows the square root transformed allele fractions. By looking at those lollipop plots, we actually see there's inconsistent distribution of anterior to posterior between the left and right hemisphere. But the allele fraction differences seems to be very consistent between the left and right lobe. Also, we found that the P1 labeled microglia seems to be a very distinct lineage compared with all cortical-derived cell lineages. Similar to rodents, the neurons and oligodendrocytes seem to have a very high correlation in terms of their allele fractions. To quantitatively show the differences we observed in the previous slide, we use this plot to show on x-axis the normalized delta between the allele fractions measured on the right hemisphere minus the left hemisphere, and on the y-axis the allele fractions from the anterior brain minus the posterior brain. As we can see here, there's a continuous y-axis change, but we can see nothing at the x-axis, which means that the left-right split in the human neocortex seems to be happening way before the anterior and posterior distribution. By looking at the allele fractions of different sorted populations in the brain, we can further infer the progenitor pool size of human neocortical underlie. By looking at those variants that distributed in both hemispheres, the earlier the left-right split, the more difference of allele fractions we would see on the left and right hemisphere. The larger the progenitor size is during the left-right split, we will expect a smaller differences between the allele fractions from the left and right hemisphere. For variants that are restricted to only one hemisphere, the larger the allele fraction is, the smaller the initial progenitor pool is during the left-right split. By estimating in those two strategies, we conclude that the human neocortical analog contains around 90 to 200 progenitors when the left-right hemisphere first split. We also established protocols to find sort out a small population of dorsally derived excitatory neurons and ventrally derived inhibitory neurons. So we see overall high correlations. We did have some examples where left and right dorsal and ventral clones distribute differently, suggesting they happened after left-right split. To conclude, our data suggests that left-right asymmetry is the first established in neurocortical development in humans, and is followed by anterior-posterior and further dorsal-ventral split. There are 90 to 200 progenitors committing to the human neocortex, and we found human cortical cells originate from both dorsal and ventral cellular populations, similar to rodents, and microglia lineage seems to have a quite different origin. We carried out the idea of cell diffusion barriers, where we see a physical barrier between the left and right hemisphere, and then a restricted barrier from the anterior and posterior parts, and also further dorsal ventral. We want to acknowledge all the members from the Gleeson lab and thank all our collaborators and funding sources. Thank you.